Okay, so and welcome everyone, and thank you so much for your time joining us today. Today, the presentation is going to be by Anna Byrd, and it's going to be about ChEA-KG. I just want to say a few words about the history of the project. Somewhere around 2010, we realized that a lot of people have been publishing cheap seq experiments in individual studies where they profile one or two transcription factors in a specific context. And so we were thinking to start collecting those publications and processing them uniformly and creating gene sets from those transcription factor targets. And then that led us to build this app called ChEA, and it was a huge success. So people can come with their differentially expressed genes and then identified prioritized transcription factors based on those past cheap-seq experiments that have been published. And we collected about 200 publications of transcription factor binding sites at that time. And then maybe 8 years later, Ali Keenan, MD, PhD student that was in the lab, her thesis project was to upgrade that system, and she integrated multiple databases of transcription factor targets, and the data, became more available. Other tools came up, so she created this version of ChEA, which we call ChEA 3, because there was also a ChEA 2 that never really got published. But ChEA 3 integrated 6 databases and was a better way, sort of like also benchmark those other tools that were competing with ChEA and benchmark them to show that ChEA 3 has like superior ability to guess their right transcription factors given a set of differentially expressed genes. And now, sort of like another 8 years later, Anna picked up the project to create the ChEA-KG, which is a next step for the project where we created this new, highly accurate and comprehensive gene regulatory network for the human transcription factor network. And then now we project the ChEA results onto this network. So Anna, please take it away.
All right, so yeah, once again, thank you everybody for being here, and thank you, Dr. Mine, for the introduction. So my name is Anna Byrd. I am a Bioinformatics Software Engineer in the Mayan Lab at the Icahn School of Medicine at Mount Sinai in New York. And as Dr. Maya mentioned, today I'll be presenting on ChEA-KG. This is a new application we've developed, which has a transcription factor enrichment analysis tool with that underlying human transcription factor regulatory network and a knowledge graph user interface. All right, so to orient you to the structure of the presentation, first I'll outline the background and our motivations for constructing a new gene regulatory network. Then we can dive into how the ChEA-KG background network was constructed. I'll give a short demonstration of how the site actually allows users to analyze the network for their own research. And then finally, I'll show two use cases, one which is characterizing the mechanisms of lung cancer, and the other which analyzes time series data to explain how transcriptional control changes over time. So we'll start with some background. So as was mentioned earlier, ChEA-KG contains this regulatory network of transcription factors. And transcription factors are proteins that control gene expression by binding directly to DNA in the promoter region of genes. And they play a fundamental role in expression regulation. And so consequently, their dysregulation is linked to disease. And we estimate currently that there are 1,639 known or likely human transcription factors. And we also know that transcription factors regulate the expression of other transcription factors to form this really complex regulatory network, kind of like the one shown on the slide here. But the composition, topology, and function of such gene regulatory networks is still mostly unknown. So there are a wide range of experimental and computational methods that have been developed to build these gene regulatory networks, but they each have their own limitations. So to facilitate visualization and storage, gene regulatory networks can be represented as knowledge graphs, or KGs. And knowledge graphs are network representations that have two main components. The first are the nodes, which are the different entities in the network, and then the edges, which show how those entities are connected. And additionally, for knowledge graphs, the edges have semantic edge labels, which give more information about the nature of the relationship. So for example, this knowledge graph, this mini graph, is showing the negative feedback loop between the transcription factors SPI1 and GATA1, or sorry, I should say positive feedback loop with negative edges. And this connection is actually recorded in literature to explain or these transcription factors are known in literature to control erythroid cell fate. So for ChEA-KG, we constructed a new GRN and we store it as a knowledge graph. And this gene regulatory network has signed and directed edges and also includes all human transcription factors. And to increase the accessibility of the network, we designed the ChEA-KG application to facilitate interactive visualization, and it is available via in the browser. So our hope is that users can use ChEA-KG to explore and learn from this new gene regulatory network that we constructed. So let's dive into the actual construction process. So first, I want to introduce two tools that were really instrumental to creating this network. And so the first is RummaGEO. This is a new resource from the lab that its development was led by Giacomo Marino. He recently left our lab to pursue his PhD at Yale. And RummaGEO is a search engine that serves almost 180,000 human gene expression signatures which are up and down gene sets that are uniformly processed from studies that have been published in the Gene Expression Omnibus. So RummaGEO gives us access to a large number of gene sets that are up or down regulated in a wide range of contexts. So a little more about how RummaGEO works. First, the studies are identified on GEO that have bulk RNA-seq data with at least 2 samples per condition. So for example, this study that I've shown here, this is the GSE ID, and it's comparing primary melanocytes, which are these FM samples at the top, against 3 different melanoma cell lines. These are the SK-MEL28, SK-MEL147, and the UACC62. So the sequence data for each of these samples is extracted, and then it's aligned using ArchES4, which is another resource developed by our lab. And then RummaGEO uses these study metadata to automatically group these samples by condition. So you can see that this has replicated the same sample groupings that I mentioned earlier based on the metadata. And then RummaGEO takes these different groups within the study and uses it to perform differential expression to create these gene expression signatures. So the site itself enables users to be able to search these gene sets. So there are several ways to do this. The first is to submit your own gene set to find ones that are similar. You can also search PubMed to find sets that are associated with the relevant papers, or you can search the sample metadata. So for example, this is an input gene set, and it'll be queried against the background database of signatures to find ones that are similar. So this is a snapshot of the results. This is actually produced by searching that GSE ID in the study, searching it in the study metadata. And this just allows us to see the signatures that are produced automatically by RummaGEO. So we see each group versus group comparison, for example, between these 2 melanoma cell lines up here, or between the primary melanocytes and one of the cell lines, and so on and so forth. So next we have ChEA-3, which is a tool, as Dr. Mayan mentioned, whose development was led by former MD PhD student Ali Kanan. And this is a transcription factor enrichment analysis tool that predicts the likely transcription factor regulators for an input gene set. So the enrichment analysis itself is based on 6 background libraries of transcription factor target relationships. And these are collected from a variety of experiments, including ChIP-seq, text mining, and co-expression. And altogether, the background libraries contain 1,632 transcription factors. So almost all known human transcription factors. And what this looks like in practice is that you can input a gene set for analysis, and ChEA-3 will rank for each of those libraries all of the transcription factors in that library based on how similar their target gene sets are to the input gene set. And then it averages the rank across all of those libraries and uses that to perform the final ranking, which you see here. And in this case, the higher a ranking, the more likely it is, or the more similar that transcription factors target genes are to your input gene set. And so this enables you to infer which transcription factors are the most likely to be regulating the genes in your gene set. So we use this to our advantage to build ChEA-KG. So first, we download the gene sets from RummaGEO. To make sure that we can infer causal relationships, we focused on gene sets that have clear control versus perturbation groups. So this is a total of 29,813 gene sets. And I kind of represented them on the right with these little bars representing the gene sets, and then the sign representing the sign of the signature. And then additionally, the asterisks indicate genes that encode transcription factors, and that'll become important later. So we can take these gene sets and then submit them to ChEA-3 to get the enriched transcription factors for each gene set. So in this case, we took the top 10 for each one, and these become our source nodes. So for each gene set, we have the 10 transcription factors that are most likely to be regulating the genes in that gene set. And if we assume that those transcription factors are in fact regulating those genes, then we can infer edges between each enriched transcription factor and the transcription factors that are in the gene set. Additionally, we can give a sign to that edge based on the sign of the signature. So for example, for this upregulated gene set, we are assuming that transcription factor 1 and transcription factor 10, just arbitrary, are actually controlling the expression of those genes in the gene set. And since that gene set is upregulated, we are going to infer that those transcription factors are upregulating the genes in that gene set. And so that's how we collect our initial set of edges. And we repeat this again for all of those 30,000 gene sets. So after this process, we end up with an initial network that has 1,560 transcription factor nodes, which is great because that's almost all known human transcription factors. But we have 373,000 edges. And additionally, this shows the distribution of in degree and out degree for the target and source nodes, respectively. And we can see that the network, based on the size and the connectivity, is far too dense for all of those relationships to reasonably be expected to be direct relationships. So we decided to prune the network. And our pruning strategy is based on the assumption that the biologically relevant interactions are going to be observed across many conditions and contexts. And we can test that because our data set, our initial data set of those 30,000 gene sets, is very large and covers a wide range of contexts. So this is a quick example where I have some very crudely drawn cells, but assume that each one of these is a different context. And we're just looking at one example transcription factor interaction here, but let's say that this TF1 upregulates TFA in 3 of the 4 contexts, and we see that we infer a downregulating edge in one of the contexts. In this case, we're going to favor the upregulated interaction as the most likely direct interaction. So in order to do this more methodically, we randomly shuffle the network edges many times. And each time we record the number of times that each individual edge is observed with that specific sign. So that gives us an edge count. And by repeating this process many times, we end up with a distribution of counts for each edge that we can use to perform a z-test using the mean and standard deviation of that distribution. And so this gives us an idea of whether we encountered an edge more times than would be expected by random chance. So for example, this upregulated edge, we might find that it is significant, whereas the downregulated edge is not significant. So we use that z-test to calculate a p-value and then filter to only include the significant edges. So after applying this process, we were able to successfully filter the network. So we removed almost 240,000 edges. Importantly, we preserved most of the nodes, so we only lost one. We still have 1,559 nodes, which is very close to all human transcription factors. And the source or targeted source degree distributions give us more insight into the connectivity of the network. So for the target degree, we see that the average transcription factor is regulated on average by maybe 75 other transcription factors. And then for the source degree distribution, we see this interesting bimodality, and more research is needed to really understand what is causing that. But this gives us an idea of each transcription factor, each source transcription factor is on average has about 300 other targets. So once we had this final network, one of the first questions we asked was whether we could identify any functional modules in the network. So we did this in two ways. On the left side, we have a UMAP, and this shows the source transcription factors clustered based on how similar their target sets are. So each of these points is one of the 700 source nodes in the network, and the points that are close together have similar target gene sets. On the right, we have and I should mention these are colored based on light and clustering to identify the different modules within the UMAP. On the right side, we have a similarity score matrix, and we originally did this for the whole network, but then we found this region of 381 transcription factors that showed the most activity or most similarity, and so that's what's shown here. And we further identified modules within the similarity score matrix by looking for these kind of lighter colored spots and use that to extract clusters from the dendrogram. So then for both the UMAP and the similarity score matrix, we take those modules of similar transcription factors and submit that to Enricher, which is an enrichment analysis tool, to find the enriched terms for each of those modules, and then we can label them with these functional annotations. And so we see some similarities between the two. So for example, we have cell cycle. There are several pathways related, or several annotations relating to differentiation and development, and then finally immune response. All right, so now that we have kind of a basic understanding of how the network was built, I'm going to show you how the site allows you to interact with the network to hopefully further your own research. So this is the ChEA-KG site. We see this really nice announcement about the seminar today, which I'm going to dismiss for the moment. And so the home screen is the single term search. So this node in red is our query transcription factor, and then the blue nodes are the immediate neighbors of that transcription factor. And from this single term search, right now we're looking at Lyle1, we can see some interesting features in the network. So each of these edges have an arrowhead in order to indicate the direction of the relationship, and then the color indicates the sign of the regulation. And we see features such as this KLF1 has a positive feedback loop. And then, for example, we can see negative feedback loops as well. And then it's kind of hard to tell, but Lyle1 also has a positive feedback loop with itself. So it's regulating its own expression. So from here, we can toggle this end node switch. And what this does is it extends our search just an extra step away from our search term. So now we can see, for example, this ADNP2 is now 2 edges away from our search term. And I apologize, this view is a little messy, but the good news is you can move everything around to clear it up. But we can also enter a end node. And what this is going to do is show us the connections between 2 transcription factors. So in this case, we have a direct connection. We see this negative feedback loop between Lyle1 and IKZF1. But in other cases, there might be some intermediate transcription factors. And so in that case, those would show up here as well. So the other main feature of ChEA-KG is this enrichment analysis tool. And this is based on the ChEA-3 transcription factor enrichment analysis. So this example gene set is actually one of those gene sets that I mentioned that were created by RummaGEO. So in this case, it's the SK-MEL28 cell line compared to the primary melanocytes. And we see the upregulated genes here, so we can submit that gene set to search. And what we see in the results are so each of these nodes is one of the top 10 transcription factors from ChEA-3. And then we get the edges from the background network in ChEA-KG. And this kind of gives you more context into how those transcription factors coordinate together to control the expression that is observed in your input gene set. It also allows you to identify important nodes within the network. So for example, we see HMGA1 in the middle here is very connected to other nodes in the network and is involved in several feedback loops as well. So that could potentially be an important master regulator of this subnetwork. While we're here, I'll also mention that for those who want to learn how to create their own gene sets to submit to ChEA-KG, we offer 2 example notebook workflows that also explain a little bit about how to use the ChEA-KG API. So the first one is starting with RNA-seq data and shows how to create this melanocyte example. And then the other one is using a bed file for those that have ChIP-seq data that they'd like to analyze. So from the enrichment analysis tool, we decided to extend it in order to provide users with more examples that could potentially be relevant to their research. So the first is the cell atlas. And both of these atlases that I'll talk about are based on manually curated marker gene sets. So in this case, we have 131 major human cell types representing 14 different tissues, which you can see here. And for each of these cell types, we queried enricher cell type libraries to find corresponding terms and extract those marker gene sets. So at the moment, we are looking at the erythrocyte progenitors in the blood. And if we switch to this View Marker Genes tab, you can see exactly what term and what library, as well as the marker genes, were used to create this network. And so then looking at the network itself, I wanted to highlight one thing, which is that we now have 2 different node colors. And all this indicates is that some of the enriched genes, these ones, or some of the enriched transcription factors, these nodes in red, are also present in your query gene set. So that is the cell atlas. And then we also have the cancer atlas. So these subnetworks represent 69 subtypes from 10 different tumor types from the CpTAC3 cohort. So we previously had identified these transcriptomic subtypes by creating a UMAP of the samples within each cancer type and then using Leiden clustering to cluster the samples. And this is based on transcriptomics. And so those subtypes are here. And if you'd like to learn more about that process or see any of the other analysis we did, you can click on this button to view the report in multiomics to targets. So let's look at lung squamous cell carcinoma, since I'll be talking about that later. And let's go to subtype 2. So once again, we see that we have several transcription factors that are also in our input gene set. Those are these nodes in red. And we see several transcription factors that are related to immune response. So for example, SP110, SBI1, so on and so forth. And so from this network, we can potentially classify or begin to think about this subtype as an immune subtype, an immune hot subtype, I should say. And so the final thing that you can kind of look at in the site is we have a help page, which just gives an overview of how to use the site and customize the network view. And then we have a downloads page as well, where you can download all of the relevant files. So that is my super quick demo. All right, so now I would like to show you 2 use cases that will hopefully provide some inspiration on how to use ChEA-KG for your own research. So the first one, we'll use ChEA-KG as part of a workflow to characterize the mechanisms of different subtypes of lung squamous cell carcinoma. So as I mentioned earlier, these are actually using the exact same marker gene sets from ChEA-KG as in the Cancer Atlas. But once again, this is lung squamous cell carcinoma from the CpTAC3 cohort, and then we identify upregulated genes for each of those transcriptomic subtypes. And so using those marker genes, first we submit them to Enricher to get kind of a baseline understanding of the enriched pathways for that subtype. And then we also submit them to ChEA-3 to identify the regulatory networks that are controlling the transcription that results in each of those subtypes. And to further kind of understand those results, we took each of the transcription factors in the network and used a new tool from the lab, which I'll explain in a moment, called the Gene Set Foundation model to make phenotype predictions based on each transcription factor. And we combined those along with the enrichment analysis results to assign a final characterization for each subtype. So quick background on GSFM, or the Gene Set Foundation model. This is a new tool from our lab. Development was led by Daniel Clark. And it is a deep learning model that predicts gene functions. So it is trained to recover held out genes from gene sets. And the training data includes thousands of these automatically extracted gene sets from RNA sequencing studies. And so it learns to predict those held out gene sets, or held out genes from the gene set. And it has also been extended to augment user input gene sets. And the paper is actually in bioRxiv right now, so you're welcome to read more about the details there. So for example, if we search KLF4, this is the results page. And so you see the automatically generated AI summaries. There are 2 that you can look at, as well as the NCBI description. And then on the bottom half of the page, which you can't see in the full results, just the top, but this is where the gene set predictions are. And this shows function predictions across the 16 libraries that are in GSFM. So using that, these are the results from subtype 1. So at the top of this table, we see the enriched terms from Enricher for the subtype marker genes. And then these are the consensus phenotype labels. So these were taken by getting the top predictions for each transcription factor using GSFM based on the model assigned confidence in the function prediction. And then we aggregate the top 10 across all of the transcription factors, and that's what's shown here. So, and then on the right, we have the ChEA-KG, or sorry, on the left, we have the ChEA-KG subnetwork. And this, again, provides more context into how those transcription factors are interacting with each other. So for example, we can see here that this ELF3, EHF, SPEDF, and FOXA1 kind of form their own module apart from the rest of the transcription factors, which is potentially interesting. And so then to actually assign the characterization, we go through the results in the table. So for example, in this one, we see neutrophil chemotaxis, neutrophil migration, granulocyte chemotaxis, granulocyte number. And so based on this, we assign, just by kind of manually assessing these terms, we assign it the characterization of inflammatory subtype. So for subtype 2, we repeated the same process. So several of these pathways are related to things like RNA processing. We have cross-link repair, protein import into the nucleus. There are some morphology-related phenotypes. And so based on this, we conclude that this is likely a cell cycle-related subtype. Next, for subtype 3, doing the same thing, we see inflammatory response, T cell activation, T cell proliferation, immune system phenotype shows up twice. And so this phenotype is related to, this subtype is related to immunity. For subtype 4, we see DNA transcription, transcription by RNA polymerase, so on and so forth, and can assign this subtype the DNA replication characterization. For subtype 5, we see several pathways related to metabolic processes. So hypoxalin metabolic process, heme metabolic process, we see glutathione, as well as this decreased urine creatinine level. And so based on this, we might conclude it's a metabolic subtype. And then finally, subtype 6, we see several terms related to cilium assembly and movement and function. And so based on that, we assign the cilia structure and movement. So just to kind of summarize all of that on one slide, here I have each of the subnetworks for each of those subtypes, as well as the characterizations that we assigned. And you can see, I think, more clearly from this slide, the insight that ChEA-KG provides, which is, for example, in the cell type subnetwork, we see this submodule of 6 transcription factors that are very connected to each other. Same thing for metabolism as well as inflammation. So it just provides that additional context to kind of guide your research. So finally, for the last use case, we'll look at how ChEA-KG can be used to uncover temporal transitions in transcription factor modules. So this project is being completed as part of our summer research training program, and it's being led by summer trainee Andrew Van Dusen. His picture's down here. And so for a quick background, changes in tissue development or tumor progression over time involve changes in gene expression. And we can measure these changes by measuring the RNA expression at different time points. So our idea is that if we submit the different time series data to ChEA-KG, we can visualize how those transcription factor regulatory networks evolve over time, and which offers insight into the transcriptional programs that are maybe driving these changes. So in this particular example, we use a study that aims to understand the effects of TRAIL treatment in triple-negative breast cancer. So TRAIL is, and apologies, this should be TRAIL, but it stands for tumor necrosis factor related apoptosis inducing ligand, and it is a cytokine that is a potential cancer therapy. It's been shown in preclinical models to selectively trigger apoptosis in cancer cells, but the efficacy has not been as high in clinical trials. And so these researchers were attempting to figure out why. And so to do this, they use this triple negative breast cancer xenotype or xenograft model, treat it with TRAIL, and then measure the RNA expression at various time points, as is shown here, to better understand the mechanisms behind TRAIL treatment and why the efficacy might decrease in a clinical setting. So we extracted the up and down regulated gene sets for each time point from this study by performing differential expression between each time point RNA-seq data and the time point 0. So we have hour 1, hour 3, 6, 12, and 24 here. So the blue nodes show the transcription factors that are enriched for the upregulated gene sets, and the red nodes show the transcription factors that are enriched for the downregulated gene set. And then, as I said, each row is a consecutive time point. And then the edges, once again, are from the ChEA-KG background network. And so starting to look at the results, first we see this NF-kappa B2 and REL-B. Both of these show up as enriched for the upregulated genes around hour 12. And these form a heterodimer in the non-canonical NF-kappa B2 pathway, which was actually found in the study to be activated by TRAIL and contributes to immune suppression, which is a potential explanation for why efficacy decreased in the clinical trials. Additionally, from the ChEA-KG edges, we see this ZNF524 transcription factor, which is relatively unstudied, and it downregulates both REL-B and NF-kappa B2. So its direct downregulation suggests it could potentially be a novel regulator of this pathway. So next, in the earlier time points, we see several early response transcription factors. These are these ones in blue. So we have FOS-B, JUN, and FOS. And then we also see several nodes that are the target of many other transcription factors. So they have a lot of incoming edges. So these are potentially of interest to begin to target this pathway and alter the expression. So in summary, ChEA-KG is a transcription factor enrichment analysis tool that places the enriched transcription factors in the context of a human gene regulatory network. This network connects all human transcription factors with signed and directed edges. And by using statistical methods to extract those edges, we are potentially uncovering new transcription factor modules and pathways. And then finally, the subnetworks can be used to identify upstream kinases as well as other potential targets. So that concludes my presentation. I thank you very much for coming. I hope that this was informative. And finally, I would like to acknowledge all the members of the Mayan Lab for their support, especially Dr. Mayan for his guidance, as well as to Errol Evangelista and Andrew Van Dusen for their work in the project. So thank you again, and I guess I can take any questions now.